Hello everyone, myself Nayana Reddy. I am studying in Sri Venkateshwara College of Engineering. In this video, I am going to explain about bioprinting techniques and materials. The topics to be covered are 3D bioprinting, basic principles of 3D bioprinting, how does bioprinting works, 3D bioprinting techniques, materials in 3D bioprinting, and conclusion. First, we can discuss about 3D bioprinting. Bioprinting is utilization of 3D printing techniques to combine cells, growth factors, and or biomaterials to biomedical parts, often with the aim of imitating natural tissues characteristics. Generally, 3D bioprinting can utilize a layer by layer method to deposit materials known as bioinks to create tissues like structures that are later used in various medical and energy fields. 3D bioprinting covers a broad range of bioprinting techniques and biomaterials. Currently, bioprinting can be used to print tissue and organ models to help research drugs and potential treatments. However, span from bioprinting for extracircular matrix to mixing cells with hydroxide deposited layer by layer to produce the tissue. Physical organ model, real organ, bioactive tissues model. These are the 3D printing and 3D bioprinting. Purpose of the printing: doctor-patient communication, interoperative negligence, performing experiments, pre-operative planning, stimulation operations, medical education, training skills. The purpose of bioprinting: cell reactions, cell therapy, drug screening, local tissue filling, tissue recognition, organ transplantation. Basic principles of 3D bioprinting: pre bioprinting, bioprinting, post bioprinting. First one is pre bioprinting. Pre bioprinting involves creating the digital model that the printer will produce. Technologies used are and magnetic imaging MRI scans. Second one is bioprinting. Bioprinting is actual printing process. Bioink is placed in a printer and deposition takes place based on the digital model. Third one is post bioprinting. Post bioprinting is a mechanical and chemical stimulation of printed parts so as to create stable structures for the biological materials. Next one is how does bioprinting works? 3D imaging and 3D modeling by biolink preparation. First one is 3D imaging. To get the exact dimensions of tissue, a standard CT or MRI scans is used. In 3D imaging should provide a perfect fit of tissue with little or no adjustment on the part of surgeon. Second one is 3D modeling. A blueprint is generally using AutoCAD software. The blueprint also includes layer by layer instruction in high details. Bioink preparations. Bioink is a combination of living cells and a base like collagen. The latter provides cells the grow and on nutrients to survive on. The complete substance is based on the patient and is function specific. Next one is 3D bioprinting techniques. Fused deposition modeling, inkjet bioprinting, laser assisted bioprinting, other techniques, VAT polymerization, extraction based bioprinting. First one is fused deposition modeling. Fused deposition modeling, also known as 3D printing, is one of the oldest process among AM techniques. According to the mechanism of printing by feeding a thermoplastic polymer into a liquefier and consequently excreting a filament, it produces the melted thermoplastic allocated as a support for bioinks. Has several parameters affect the property of parts manufactured by FDM. Researchers try to focus on key parameters to optimize the fabricated parts and finally get the combination of them. These are the materials, variable parameters, and chemical properties. PLA. In variable parameters, there will be layer thickness, infill density, post process heat treatment. In chemical properties, shear stress. Material PLA. Variable parameters will be deposition organization, layer thickness. In chemical properties, tensile strength, impact strength. Material APPS, variable parameters or temperature profile, temperature variations, chemical properties or 3 point bending test, thermal analysis. Material peak, temperature variations, chemical properties or mechanical properties or tensile comparison, tensile strength. Second one is stereopalography. In the 19th century, a high printing quality technique based on polymerization of high sensitive polymers was developed. The main mechanism of this technique is based on utilizing UV laser and a directed mirror array to stick out the light beam on the surface of the liquid photograph design. This is repeated in each layer along Z directions to set up the 3D part. The main disadvantages of SL in UV light source is which is to the biocells and cause skin scanner. Third one is inkjet bioprinting. Inkjet based bioprinting is non-contact process by using 2D desktop inkjet printers. In generally, they are classified as thermal inkjet bioprinting, electrostatic bioprinting. A voltage pulse is generated to inkjet bioprinters to generate droplets by applying between a pressure plate and an electrode. This is extrusion based bioprinting, also known as direct writing, has been widely engaged in biofabrication and tissue engineering in recent years. The distribution of bioinks or as a stand widely takes place based on the two main mechanisms. One is mechanical force, another one is poisonous force. In fact, it contains of a system placing on a robotic stage and controlled by a stage controller. Next one is laser bioprinting. The bioprinting techniques originally developed for writing of a metal successfully applied in bioprinting of nucleic acid such as DNA or organ cells. The first laser assisted bioprinting was developed in 2004 to convey of biological patterns to substrates in order to print biomaterials with high resolution. As explained in previous sections, bioinks or those cells in liquid or gel solutions depositing over the metal film support. The metal film is in variations along with the incident layer light, which is leading to removal of bioink droplets. Last one is VAT polymerization. VAT polymerization technique releases on polymeric solutions that are based on a laser solidification mechanism. The main types of VAT polymerization techniques have been introduced so far: digital light processing and that are systematically illustrated. Seventh one is other techniques. Numerous studies have focused on development of 3D bioprinting techniques. To date, many types of techniques have been successfully developed and applied in field of bioprinting. However, among the represented techniques, other related methods are being widely utilized in this field. This is one. The major technique is generated of co-cultural, which is not needed to be sincerely necessarily applied. A systematic diagram of magnetic bioprinting and biopolting is indicated. Materials in 3D bioprinting, synthetic polymers, homogeneous is degradation and printability are the main important factors for biomaterials with applications in 3D bioprinting. In general terms, biomaterials are natural and synthetic substances being used for repair or even replace any organ of a body. Next one is bioinks. The term bioinks is an important issue in organ bioprinting and tissue industry. Bioinks encamps biomaterials and living cells in a cellular matrix environment. In comparison with traditional 3D printing materials, bioinks must have non-toxic bioactive components as well as a print temperature below physiologically temperatures. According to According to bioinks are classified into four main groups based on their roles: functional bioinks, support bioinks, and structural bioinks. Varieties of materials are being used in bioinks to perform 3D prints. of structures such as polymer. This section describes the strength and limitations of these materials in bioprint 3D printing. Conclusion: additive manufacturing has been heavily applied to tissue engineering over the past decade. Bioprinting enables the production of the with homogeneous distribution of cells throughout the scaffold. Organized distribution of different cells types can be positioned within the supporting materials. Future developments are now concentrating on the combining of techniques to work in complementary fashion to optimize the process of creating tissue structures. Thank you.